Hello, I'm Adrienne Fairhall, and I'm at the University of Washington. I'd like to welcome you to this Brain Prize webinar in honor of this year's Brain Prize awardees, Larry Abbott, Haim Sompolinsky, and Terry Sejnowski. This is the first of a series of 6 webinars in the theme of the area of our awardees' general research, neural circuits of the mind, frontiers of computational neuroscience. And these seminars will be on topics including spatial navigation, learning and memory, sensory cognition, decision and behavior, and large language models and human language. If you'd like to find more information about those are on the Brain Prize website. So it's a delight to introduce today's speakers, the honorees. The first, Larry Abbott, is a professor at the Zuckerman Institute for Brain, Mind, and Behavior. He's a co-director of the Center for Theoretical Neuroscience and of the Kavli Institute for Brain Science. Haim Sompolinsky is director of the Center for Brain Science at Harvard University. He's also a professor of physics and neuroscience at the Edmund and Lily Safra Center for Brain Science at Hebrew University. Terry is a professor at the Salk Institute for Biological Studies, and all 3 are directors of Swartz Centers for Theoretical Neuroscience at their respective institutions. So none of them wanted a lengthy introduction, but I'm going to just give a little factoid about each. So all 3 came through from physics, and I went back in time to find out the moment of transition for each of them from physics into neuroscience. So Larry came from particle physics and cosmology, and his first paper in neuroscience was in 1988. It was called Domains of Attraction in Neural Networks. Haim is a statistical physicist, and his first paper in 1986 was neural networks with nonlinear synapses and static noise. Terry came from astrophysics and even published in Gravity Waves. His first paper was in 1976 on global properties of neuronal interaction. So these 3 scholars, through their amazing scientific contributions over many decades, their teaching, both at their own institutions and in summer courses, their leadership as center directors, and also through advocacy in the Brain Initiative and other mechanisms, have really inspired multiple generations of researchers to apply mathematical methods to understanding the brain. So on behalf of all these generations, I would like to say we could not be more delighted at this recognition to the awardees. So congratulations to all 3. And without further ado, let's start the seminars. So Larry Abbott is going to be talking about a sense of self. So turn over to Larry.
Thank you, Adrienne. My talk indeed is about how we get our sense of self. The sense of self I'm talking about really consists of 2 parts. How do I know what my effect is on the world as opposed to the world effect on me? For example, I know my slides are advancing because I push a button and something happens on the screen. Now, if slides were advancing without me pushing the button, I would know somehow, somebody's hacked into my presentation and I'm not in control anymore. So this idea of understanding the effects you have from your own action as opposed to the effects that the world imposes upon you. Now, another aspect of it is my place in the world and my sense of the objects around me. So, for example, I'm sitting here in my office. You can see a door behind me. I know where the door is. And even if, I turn my body or something, the way the door appears to me changes. It's now at a different angle. But I certainly understand that the door in the room is still where it was. So I sort of have a dual coordinate system. The world that I see through my eyes, which move around with my body and turn with my head, and the world as I understand it to exist outside of myself with its own coordinate system. So I'm going to talk about the neural basis of these very fundamental aspects of being a creature in the world and understanding what the world's all about. But I'm going to do it in much simpler systems where we can really figure out the neural basis of this happening. So the first topic, the effects of yourself versus the effects of the world, I'm going to discuss in an electric fish that you see here. And the second topic, the construction of a coordinate system for the world independent of yourself, I'm going to discuss in flies. And you'll see how in detail the neural circuits develop these fundamental senses of the self and the world. Let me start off with the humans involved here. This is all collaborative work with experimentalists. In particular, it's collaborations with Nate Sawtelle on the electric fish and Gabby Mayman on the fly. Everyone on this slide is an experimentalist working in these labs, except for Solomon Mueller, who did the theory work on the fish thing. But I could not be speaking without all the beautiful work of the people that you see on the screen here. Okay, so let's start with the fish. The fish is called the elephant nose fish for kind of obvious reasons, because it has this funny kind of chin on the front of it. And that fish is covered, its skin is covered with electroreceptors, extremely sensitive electroreceptors that are able to detect very weak electric fields from other objects in the water, for example, other creatures in the water, for example, prey, bugs that the fish might want to eat. Now, this is a passive system. It's not what makes this an electric fish. In fact, there are non-electric fish like sharks that are very good at this as well. The thing that makes this fish an electric fish is it has an organ in its tail, and several times a second it will put a shock into the water, and that shock will create an electric field around the body of the fish. Now, that is used for communication between fish, and it's also used for what's called active electroreception. But I'm going to talk about the passive system with these very sensitive electroreceptors. And for the passive system, this is a tremendous source of noise. It would be a little like if I flashed a strobe light in my face but was trying to look at my slides. And so the fish has got to get rid of that overwhelming self signal and distinguish the non-self signal. So I hope you can see how this is an example of an animal that has to distinguish between self and the world. Okay, so the electroreceptors on the skin, of course, receive a combination of these two electric signals, the prey signal and the self signal. The prey signal I've shown below is smaller in amplitude. And of course, that's what the fish is interested in. It wants to find that prey and eat it, for example. The other signal, these big spikes, that's the self-generated field of the fish. And it is not interesting. It's totally predictable. The fish knows it's making it. It doesn't do the fish any good in this context. And so the fish is going to get rid of that. And the key to this is that the fish is generating the field itself. So it knows that that electric field should be coming, and that predictability allows the fish to cancel it out. Now I'm going to consider a particular cell type that's kind of in the middle of the circuit that does this task. It's called an MG cell. It looks like this. And the way it works is the following. The electric signal from the skin comes in. And again, that's the combination signal that includes the prey signal as well as the self signal. But then it also receives inputs that tell this cell the fish just fired an electric organ discharge, and it should be expected that that self-generated field should be coming in through the skin. Now, importantly, the synapses that connect this information from the rest of the brain to this cell are plastic. And what they do over time is to construct a prediction of the predictable part of the signal, which of course is the self-generated part. This system cannot predict the prey part because that's not under the control of the fish, but it can predict the self-generated part, and the plasticity kind of automatically results in a cancellation. So the upper signal generates what's called a negative image. It's a duplicate of the self-generated electric field, but with a flipped sign. And so these should subtract each other. And what you might expect is that they subtract from each other, and what's left is only the prey signal. That was originally how this cell was thought to operate, but actually that's not what happens. Instead, the signal that's coming through this bottom pathway never makes it to the output of this cell. It just gets blocked. And instead, what gets transmitted then is just this cancellation signal. So the full signal is not. And so this cell is actually an identifier of self. From this mixed signal, it pulls out the piece of the electric field due to the self. Now, the question here is, how did this happen? Because these are just two synaptic inputs to the same cell. And yet what I'm telling you is that one of them goes through normally to the output and the other does not. I'm not going to take the time here to explain exactly how this happens, but we made detailed multi-compartment models of this cell. You can see the different regions here. At the top is where the self-generated EOD internal signal comes. At the bottom, the external skin signal comes. The red is the axon, and the brown is sort of where the self-generated plasticity occurs. But anyway, what happens is that the cell cannot actually transmit the skin signal, and as I mentioned, only transmits the predicted signal. A critical component of that is that the model required that this input not come in directly as just an excitation into the MG cell, but it required a very kind of funny, non-intuitive double inhibition. So rather than exciting the cell directly, the skin signal inhibits one cell, which inhibits another cell, which inhibits the MG cell. So it's like two minus signs making a plus sign. So this was odd, but as I say, it was critical to assuring that the only transmission occurring from this cell is the upper pathway, if you want, the negative image self-prediction. Now, this is kind of looked like a crazy feature of a model, but fortunately, in the interim, we have, in a big collaboration involving multiple teams, we have developed a connectomic understanding of this circuit. So down to individual synaptic levels. It's a piece of the circuit, but it includes all the cell types, and in particular, the MG cell and all its inputs. And if you kind of look at that structure, it kind of looks like a mess. Everything is on top of each other. This is the MG cell here. This is the afferent input from the skin. And here is the first inhibitory cell, the second inhibitory cell. But just from looking at it, you would think that everybody's kind of connected to everybody. But at the individual synapse level, what you realize, in fact, it's connected just like in the diagram below, which we surmise from the model. So let me just finish by telling you how the whole circuit works. We have this initial step that identifies the self signal, but that feeds into an output layer. And that output layer also gets the skin signal, but it gets it flipped in sign. It's an inhibitory input instead, so it gets kind of the inverse of the signal. Then it gets this self signal from the MG cell, and now the cancellation takes place. These two sum up at the output, and the only thing left is the praise signal. So it's a two-stage system, what used to be thought of as a one-stage system, and after a two-stage system. And the model also makes some predictions about all this wiring here. You notice, it has to be an inhibitory input. The inhibition has to go to the right output cell. And indeed, looking at the connectome, all of the predictions of that model have been verified through the connectome. So the upshot of here is, at least within this circuit, we have a very detailed understanding of how the fish distinguishes what it produces in the world as opposed to what the world produces in the form of prey. So now let me move on to my second topic, the place in the world and the relationship, and talk about the fly. So here's a fly out on a sunny day. It turns in the world, turns back. And the first question is, how does it know it's turning in the world? How, it sees the world turning in its visual system, for example. But how does it know that there's a stationary world and that it's itself turning in the world? This sense of self in the world. That was discovered in a beautiful paper by Sejnowski and Haim a number of years ago. And it consists this round object that you see in the center of the fly brain called the ellipsoid body. The ellipsoid body is a circle, so you can parameterize it by an angle that goes from 0 to 360. And what Sejnowski and Haim found is a hotspot of activity within that structure. And that hotspot obviously encodes an angle, depending on where it is. I believe Haim in his talk will talk about the network responsible for this. I'm not going to do that, but I'm just going to tell you about the consequences. The consequences of this is that that dot in the fly brain can be considered a direction denoted in the fly's brain. And when the fly turns in the world, that direction turns in the fly's brain. So in other words, the fly has in its brain a little compass that tells it it's heading in the world, and that denotes the fact that it's the fly turning in the world, not the world turning around the fly. Now, I want to talk about a little more than that, and that is a whole coordinate system in which objects and events and things like that in the world can be put into world coordinates and not just the coordinates of vision or other senses of the fly. So to denote that, I'm going to use wind as an example. So wind is detected by the antennae of the fly, which you see in this diagram out on the front. And they are projected onto axes 45 degrees in each direction relative to the fly's head axis. So there are neurons that just report how strong the wind is blowing in those two directions. Now, if the fly turns in the world, of course, the way the antennae interact with the wind changes, and the fly will know something has changed through that system. But it won't be able to distinguish from that system whether the wind changed its direction or the fly turned in the wind, which is the case I'm depicting in this schematic. So how does the fly denote the fact that the wind is actually constant in the world despite its changing relative to the body? And the way it does this is to construct in its head a coordinate system and link that coordinate system to the world. In other words, in this mentally derived, internally generated coordinate system, these axes turn when the fly turns. So we have a coordinate system that acknowledges that the fly is turning in the world. And if the projections of the wind onto these two sensory axes in the middle picture are projected onto the internal axes in the fly, what you get is an invariant representation of the wind direction. So now the fly has in its head the fact that out in the world, the wind is constant, even though my sensory system is picking up different signals as I turn. Now, how does the fly do this? Very simply, it couples this coordinate system to the heading direction signal that I just talked about. So when the heading direction signals, it drags along these two axes, and you get a beautiful rotating coordinate system. Okay, so how does this really work at the neuronal level? To do that, I have to introduce two other regions in the fly brain, what's called the protocerebral bridge that you see here, and the fan-shaped body, and tell you a little bit about them. So the ellipsoid body, I already told you, it's a circle denoted by an angular label, and it has activity in it in cells that look like this in the electron microscope pictures, and gives the heading signal. All right, that is transmitted to the protocerebral bridge, and the protocerebral bridge, although it looks like a handlebar kind of arrangement, is actually anatomically or circuit-wise another set of two circles. So there are now three circles in general that can all be denoted by these angles. And at that stage, the heading signal gets handed off to another set of neurons that I've depicted one of them here. And those neurons have a very interesting property. You can almost see it with your eye in these dendritic connections that you're seeing sprinkled across the structure. If you look at the density of those or the synaptic counts, you find that they're arranged in a sinusoidal pattern across the structure quite accurately. So these cells act like filters, and they modify the bump shape, the thing that I've denoted by the blue dot, the hot spot of activity in the fan-shaped body, and they turn it into a sinusoidal shape. That sinusoidal shape is then input into this next set of cells, what are called PEN cells, and they're going to take that signal down to the fan-shaped body, where really the navigational decisions and computations of the fly take place. The fan-shaped body, although it looks like a fan, is actually another circle. Its circuit structure is, again, circular, and so it too can be denoted by an angle. And so this whole central complex, what it's called, is really consists of these 4 circles. Okay, I mentioned now that the delta 7 cells, these intermediate cells, turn this signal into a sinusoidal shape. And that's how the cells I'm depicting now, these PFNs, receive it and transmit it down to the fan-shaped body. Now that means that the peaks of these sinusoids are linked to the angle of the heading. And so the heading angle has now kind of been reproduced twice and represented as a sine wave. And I'll talk to you later about how that sine wave is then carried down into the fan-shaped body. And this is the topic of our work and a number of other papers on this subject. Now, the importance of sine waves is that they can represent vectors. The fly is obviously very interested in making this sinusoidal shape because it has a cell type devoted exactly to this. This is something that a number of authors have noted, that if you have a vector in 2 dimensions, it can be represented by a length and an angle. But that angle can also be represented by the phase or the peak value of a sinusoidal waveform, and the length can be denoted by the size or amplitude of that sinusoid. So in other words, we're representing a vector by a function of an extra variable theta. And that variable, in this case, is the variable across the array of cells whose activity is mapping out the sinusoidal shape. So in other words, if I go back to this diagram, the angle that we're introducing to get this functional representation of a vector is just the angle across these structures that I've already introduced. A nice thing about phasors is that basically any computation that you can do with vectors, you can also do with phasors. For example, if I have a phasor like this representing the red vector, and I have another phasor representing the blue vector, if I want to add those vectors, all I have to do is add the phasors, and I will get another sinusoidal shape that represents, in this case, the purple vector. All right, so how does this all work? What I've told you so far is that these PFN neurons, and by the way, they're going to be the neurons that build the coordinate system, they are linked to the heading angle such that if the fly turns, they shift their phase across these structures in concert with the turning of the fly. Now, I've mentioned that the sinusoidal shapes represent vectors, and so what you're getting here is 2 vectors that rotate with the fly and provide 2 more copies of the heading if you want. Now, in order to show you the trick that these cells use, I'm just going to show you one of these neurons rather than the whole set, which is what I've been showing thus far. So these neurons get their heading here, they get sensory input here, they put their output here. But if you look at where the output goes, you would conclude that they sort of missed their target, because remember, there are angles across all these structures. And if you look at what angle matches the heading to the output should really be there. And instead, it's shifted, and it's shifted by 45 degrees. If I showed you one of these cells on the right side, it would also be shifted by 45 degrees, but the other way. So in other words, what I'm telling you is when these neurons project this signal down to the fan-shaped body, they shift it. So instead of being 2 identical sine waves, it becomes sine waves that are shifted apart. Now, the rule about vectors is that when you shift the phasor, you rotate the vector. And what you should be able to see is that in shifting that, this cell has made an orthogonal coordinate system. And further, that orthogonal coordinate system rotates with the fly. So the only thing we have to add then is the fact that sensory data like wind gets projected onto these vectors. And that has been shown experimentally, that sensory data changes the amplitude of these activity patterns. And that corresponds to shifting the length of these vectors. And then if you just add them up, you've got your world representation of, for example, the wind, which is what I started with. So we know cells downstream of these PFN neurons that do the sum and therefore represent the structure, these vectors in the world, in world coordinates. So just to sum up, I've generated for you this rotating coordinate system that gives the fly its sense of the world. And I hope I've given you the sense of how nervous systems, in a considerable detail that we can obtain for these animals that I've discussed, how they structure and build representations that allow them, in the case of the fish, to understand the difference between what they do to the world and what the world does to them. And in the case of the fly, understand how events and sensory things that happen in the world can be mapped into the world coordinates beyond the self. Okay, thank you. I will stop there.
Thank you, Larry. So maybe I'll start with questions. So I guess what you've shown us really beautifully is a couple of examples of mechanistic models that are heavily based on connectomic information, which is maybe a point we can come back to later in the discussion. I was wondering, in the first example, you set up the original sort of cartoon model for what the MG cells are doing in a way that made a lot of sense to me, right? That you had a cancellation, whereas the model that turns out to be the case doesn't seem to make a lot of sense in the way it works. Do you have any ideas about some sort of normative principle that can explain why one would want to carry out the computation the way that it seems to work?
Yeah, that's a great question. We weren't expecting this two-stage thing. We expected two-stage of identical processing. We're still working on this, and the connectome is providing us a lot of information. I believe the reason is that MG cells are highly recurrently connected. What I didn't talk about is this system works in a kind of an on pathway, off pathway. And I really described only the off pathway. Those pathways, the learning is very different in the two of them. And we believe the recurrent system, the recurrent circuit of MGs, allows the calculation to be done kind of cooperatively between the two before you go to the output and really split it. So that's our current hypothesis, why it's done in this way, which is a bit counterintuitive.
Okay, thanks. Terry, I think you're muted.
Okay, I have a quick question, Larry. That was a beautiful presentation. In the second talk on the fly, it sounded like it was all hardwired. Is there any plasticity in that system?
Yeah, in the stuff I described, the only believed plasticity is in mapping the visual world onto the compass system. Clearly, I'm in my room, therefore I'm using the room, the features to orient. So that is plastic. But the rest of the system is not. I believe where the plasticity is, and we're working on this now, is once you form these sensory vectors, you might want to remember them. And then you're going to have a plasticity. And in fact, I really think a big point of this thing is memory. So for example, I can keep track of things in my room, even if I turn, I just look at them. But if I want to remember where something is, I would constantly have to update, right, as I turn. It's just not a viable system. So I think a lot of the purpose of this transformation and putting things in the world is memory, and that's where we're working right now.
So may I ask a quick question? So first of all, about a follow-up, you're talking about memory. Do you mean long-term memory? The fly, you think, has a long-term memory of landmarks in the environment?
So we actually know, this is work with Vanessa Ruta and members of her lab and a student with me named Minnie Sun. We've established that in plume tracking, so flies go in and out of a plume, they definitely, for example, remember which side the plume they're on. And they remember, what's long term, certainly for minutes. They don't, we don't know how long term. So yes, and we believe that occurs between neurons that represent these vectors and synapses onto other neurons that innervate them. So that when those innervating neurons get activated, a memory of a direction gets reported to the fly. We don't know that for sure from the experimental data, but that's our model of it, and the model works fine. So we just have to see if it's true.
Thanks.
Okay, I think we'll move on to the next talk, which is Haim Sompolinsky, how the brain generates a mind. Thanks, Haim.
I would like to talk today about motifs for the generation or the emergence of the mind from the brain circuits. So what do we mean when we say that an organism has a mind? It's of course a philosophical, conceptual question, but broadly speaking, I think we refer to a mind or to organism that has a mind, an organism that has internal states, mental states that are situated in between the sensation and the action, the behavior. So an organism that has a rich repertoire of such mental states, we call that organism having a mind, and those collection of states, we refer to them the mind. So the question is how actually the brain generates those states and computes with those mental states. So these classes of mental states cover many mental functions. Perception, thoughts, desire, intention, deliberation, decisions, emotion, moods, imagination, and memory. All these are examples of mental states. So I listed several characteristics that in my mind are typical of those mental states. First of all, they are representational. At least most of them, they have content, or we attribute to them content which has to do with the world, which has to do with the self, as Larry beautifully explained, the need to have a representation of self in the world and the world relative to the self. So that's one characteristic of mental states. Another one is that they're stable. Because they are in between sensation and action, they span time, and these time scales can be working memory during performing a task, or it can be long-term memory, which can span the lifetime of an animal. They are generative. They are generative in the sense that they're not only reacting to the stimulus that comes in at a given time, but they are actually generate representations that we can think about them as predictions, we can think about them as planning future action, or imagining or simulating the world, etc. Another characteristic of many mental states is the fact that they are discrete. They categorize the world. They take input, which is typically continuous streams of stimuli, either visual, auditory, and other senses, and they form a representation in terms of discrete categories like objects, concepts, words, rules, logical rules, reasoning rules, action types, etc. And finally, they are spontaneous, or some of them at least are spontaneous. The emergence of these states not necessarily linked to a particular task or a particular event in the world. This spontaneity in terms of mental states, we talk about mind wandering, creative exploration, switching between states in a so-called spontaneous fashion, etc. So what I'm going to give you briefly an overview of the accumulating understanding of some of the underlying brain mechanisms of these mental states and these properties. I'll start with historical perspectives, and for each one of them, I'll comment briefly on what are ongoing questions and some more modern or future outlook. Okay, so let me start with the stability of mental states. So the stability, as I already mentioned, comes into time scales of working memory, maybe seconds, maybe minutes, and long-term memory for which we can span many years. Okay, so our current understanding of the stability of mental states is that they are carried by persistent neural states. These are active. These neural states are neural activities that persist long after the sensory stimuli have been turned off. And this leads to the notion of attractor neural networks. So from a dynamical neural circuit perspective, neural circuits in which the activity converge over time into one of several persistent states, we call them attractors. These states we call attractors, and these circuits that generate such states, we call them attractor neural networks. Now, the properties, the number of those states, the nature of those states, their meaning, their function, all these depends on the specificity of the synaptic connection, the properties of the neuron, and the external inputs. So here in the right-hand side, you see a diagram in the state space of the circuit and neurons in the circuits. So each point here is a state of the circuit, and as the flow show, the dynamics of attractor neural network is such that you can dissect the flows into basins of attraction, wherein within each basin the trajectories converge into the fixed point or the attractor state, which correspond to that basin. So the dissection of the state space into these basins of attraction and the corresponding attractors, this is the characteristic of attractor neural networks. So how does this relate to a function, to memory in particular? So that's the topic of the Hopfield model. Is the pioneering paper in 1982, and the topic of the Nobel Prize in Physics just recently announced. So the Hopfield model is a model of long-term memory, of associative memory, but it has ingredients of both long-term memory and working memory. So the memory encoding is lasting changes in synaptic connections that make the memory states attractors of the network. That's the encoding by long-term changes in the synaptic connections, which encodes the memory. But then the memory retrieval is the process in which, given an initial cue afterwards, the network state converges to the relevant memory states and remains stable because it's an attractor, remains stable for any further processing, a function, a working memory function. So the encoding is the function of long-term memory, and the process of retrieval is the working memory aspect of that function. So in computational language, we'd say this is a pattern completion because the retrieval takes a partial information about memory and completes it by the convergence to the attractor. So that's the basic idea. And now the question is, what are the properties of such model? What are the limitations of such model? And for this, we need a theory. And the theory first described by a paper. Actually, my first paper in neural network is 1985, together with the late Daniel Amit and Hanoch Gutfreund, where we used the statistical mechanics spin-glass theory to generate a qualitative and quantitative description, characterization of the Hopfield model of associative memory. And the most important part of that theory is what's known as phase diagram. That's a classical example of how theory is illuminating the properties of collective computation in neural networks. So in this case, here's two-dimensional plane. In the x-axis, the parameter is p over n, where p is the number of neurons and the number of memories, stored memories in the network, and n is the number of neurons. And in the y-axis, the t over w, t is the level of neural noise, and w is the strength, mean strength of individual synapses after storing in them p memories. And as you can see in this phase diagram, the phase diagram is now dissected into three states. The memory state, which is the state where the system is below capacity, so the x-axis is to the left, and relatively low noise or strong synapses. So we are close to the left origin 0. So that's a memory regime. Everywhere here, the retrieval of the memory is almost perfect. And then if we are below capacity moving to the left, or the intermediate level of noise, we are in a spurious regime. The spurious regime is characterizing a network which still converges to attractors, but those attractors are typically not the memory state, but spurious states. There may be a mixture of several memories, which you can think about this as kind of a confused or hallucination or made up memory. And then finally, when the noise level is high, then there is no actually retrieval in the sense that the network states keeps wandering around and doesn't converge to attractor. So this is an example of the power of theory, and particularly statistical mechanics theory. It is rigorous in the limit of large network. It's quantitative, it's precise, it's distinct regimes. So when we talk about emergent properties or emergent computations, this is the type of emergence that we mean. We mean a very distinct properties depending on key parameters. It tells us about the robustness and the vulnerability of the model. It also gives hints for potential compensations for neurological deficits. For instance, if the circuit loses neurons, then P over N becomes large. Then you're moving towards the dangerous regime of spurious memories. However, if there is a way to globally increase the strength of the synapse, then you can move down here and still be in the memory state. So that's this kind of suggestion that come out of the model, of the theory of the model. And then finally, the theory also predicts rather substantial limitations of that model. And the limitations, the most important limitation is that the model works well when the memories are relatively unstructured. And that question of how to move beyond storing sort of random patterns as memories to storing episodic memory with rich content has been an ongoing problem and challenge for the last 40 years. And let me give you a taste of where we would like to push the theory for long-term episodic memory. And in this particular case, it's ongoing work with Ila Harami and Kazuki Irie at Harvard, trying to use large language models for episodic memory. So I'm not going to discuss the particular mechanism, it's still ongoing, but I want to show you what type of functionality we would like to have from such circuits. So imagine we look at the left as an encoding memory. So it's a story. Friends and family were at my house in New Hampshire for my 12th birthday, as we played charades, a sudden knock on the door, etc., etc. So you can see here, it is not a random pattern, strong richness of content. And what we are aiming at is to take such systems and give it a retrieval cues like my 12th birthday, New Hampshire, Lilycam, etc. And the retrieval memory, this is just an example, retrieved memory will actually not reproduce necessarily word by word the encoded memory that's non-realistic and not even desired, but actually the semantic content of that. So this is the type of functionality that we would like to have, and we basically would like to bridge between the simple circuit of the Hopfield model to a much richer circuit similar to large language models that are rich in semantics and memory. Okay, so let me move on to the generative property of the mind. So predictions, planning, imagery, current understanding of those of these properties are built on neural network that are attractors, because those generative processes are typically working memory functions. But in this case, the attractors are not discrete points, but continuous. So they're called continuous attractor manifolds. And those continuous attractor manifolds are capable of generating states, which we can think about them as hypotheses about what is out there in the world, and holding these states and updating them in working memory. So the simplest example of that is the ring network. And this ring network is as shown here in the upper left, is neurons that are organized along a ring. So each one of them has an angle, and the connectivity between them is a function of the difference between the angle as shown here on the top right. And the connection, the functional connectivity is such that it's kind of Mexican hat. Neurons that are nearby have similar preferred angle. They excite each other, and otherwise they inhibit each other. So there is a structured organization of excitation and inhibition in this network in this way. And under appropriate conditions, this network generates not only one state. When in the absence of any selective input, in the case where the input is completely uniform, still the network develops a repertoire of bump states, as already mentioned by Larry in the case of the fly, and these bump states can be centered around any position on the ring. And here is an example: 3 different initial conditions will converge into the same bump shape, but in different locations. And again, in state space, you can see here, again, attractors, but now it's not discrete points, but it is organized in a circle, and any initial condition will converge to a point on the circle. Now, interestingly, when there is an external cue, like a visual cue or otherwise, then this external cue is basically pin the bump states along the direction of the external cue. So the external cue are not generating the state, but actually select the states from the repertoire, from the attractor manifold repertoire. And this type of circuit has been suggested for a variety of functions in the brain. Our original work was with visual orientation, spatial working memory, later on generation of movement directions, and in particular the head direction by Zhang in 1996. And as already mentioned by Larry, the remarkable work by Sealing and Jayaraman and others have shown a remarkable realization of that hypothetical model in the case of the fly, and in the navigational system or the heading system of the fly, and in this particular example of imaging of the bump activity. And in this case, you can see how the bump along the ring is following the external cue as the external visual cue is moving around. But importantly, these bump states appear also when there is no, in the darkness, when it just tracks self-motion cues, and also persists in darkness when animal is not moving at all. So this is just a remarkable validation of the concept of ring attractor manifolds. And then subsequently, connectomic work, which already Larry mentioned, shown a remarkable structure in terms of the functional organization of the neurons, or rather the connection between the neurons, precisely like predicted by the ring model. Okay, so this is one dimension. This is processing an angle, but the same attractor manifold concept can be extended to higher dimension, in particular to 2-dimensional spatial self-location. And that work was motivated by the remarkable observation measurement by the Moses and originally in 2005 about the grid cells, where a single cell in the medial and triangle cortex is firing when the position of the animal is at one point along the ring. So Burak and Elafit and others have suggested that the mechanism for such a grid representation is essentially a two-dimensional extension of the ring model. In this case, the connectivity again is excitatory or effectively excitatory for neurons which are nearby preferred phases, two-dimensional phases, and inhibiting otherwise. And again, in this case, the extension of a one-dimensional ring is to a two-dimensional torus. And again, it means that every position on a torus is an attractor of the network according to this model. Okay, so this is the toroidal tractor manifold proposed to explain to be the underlying mechanism for the grid cell system. And remarkably, more recent work has actually confirmed the presence of a torus attractor manifold. This is recent work where a few hundred neurons from a single grid models were processed by the nonlinear dimensionality reduction and shown to be lying the activity of the population activity be lying on the structure of a torus. And again, this happens also in a variety of conditions: open field foraging, walking on a circular maze, REM sleep and slow wave sleep, etc. And that's important because the functionality, according to that hypothesis, the functionality of those manifold states is that the state has a characteristic shape as a population pattern of activity, but the position or the selection of the states and the update of the states is done, the update of the state location, either along the ring or along the torus, is representing or is the result of updating from a variety of sensory input. And it can be even done by self-motion, or it can be spontaneous in sleep, for instance. Okay, so this is one open challenge is how can we take this and extend it to beyond one or two dimension, beyond navigation, to a richer contextual cognitive map. And this is one direction. That potential direction is inspired by generative AI with diffusion model and is ongoing work by Bin-Su Wong from Harvard. And just give you an example of where we would like to be. And this is a diffusion model in the context of machine vision or machine generative model for generating visual. Here's an example of one network that starts from multiple initial conditions. So each square is the same network, but in different random Gaussian initial condition, and they are converging to a particular phase. So a rich manifold this is the type of here on the right, bottom right, the type of recurrent neural network that is capable of generating, if you can call it, a phase attractor. So we would like to build biologically plausible models that are able to generate attractor manifolds that go beyond a navigational or spatial coordination. Okay, so I talked about attractor manifolds and their function. I want now to go to perception, and particularly to the question how the brain generates discrete categories out of the continuous stream of sensory input. Okay, so one particular classical example is the visual system, and objects in the visual system are known to have many different physical instantiation, different background, different scale, different position, different orientation, color, etc. And it has been proposed that part of the visual hierarchy, the function of the visual hierarchy, is to be able to perform this perceptually invariant task, namely to categorize this continuous stream of physical variability in terms of discrete categories like cat, dog, mug, and hat, and so on. Now, interestingly, even in the top layer, the final stage of the visual hierarchy, which is on the IT cortex, if you look at individual neurons, they are still not invariant. There are no uniquely cat neurons or dog neurons or hat neurons in IT cortex. So the neurons are still sensitive. The response of the neurons are still sensitive to the physical features of the image. So in what sense we can say that a category emerged in that system? So here, a recent work by us, us means in this case Soo-Yoon Chang and Dan Lee and Ben Sorcha and Suad Ganguly and Uri Cohen developed the idea of object manifold. So we are looking now at the state space of the system, so this n-dimensional state space. Now each point again is the response of a neuron of the entire population into one particular stimulus. And the idea is that representation or the discrete representation of the categories is realized by a compact geometry of the manifold of the physical states that corresponding to a category, like a dog or a cat. So if these manifolds are nicely separated, are small in size, small in dimension, then they are amenable to further processing for any function that requires information about object identity rather than physical variables. And this is an example that actually we're using deep neural network to test this model. So here we're to show it's a deep neural network mimicking or modeling the actual visual hierarchy. In the x-axis is the position of the layers, starting from the left input to the top output to the right, and you have three graphs here. One is showing the degree of separability. Another one is the radius of each one of the manifolds. Another one is the dimension of those manifolds. And as you can see, there is, for instance, the separability. There is from the retina to the IT cortex model. There is a persistent incremental improvement in the representation of this manifold. But in the last stages, there is a really a nonlinear improvement. And we can say that the emergence of object manifold occurs at these stages, both in terms of separability, but also in terms of radius and dimension of this manifold. Now, this is for vision. And I want just to give you a taste of where we are heading at. We want to know whether the concept of manifolds can also go to beyond vision, to higher level functions, and in particular to language, and in particular to words. So we are collaborating. This is work with Shane Chang and Shane Shai-Li and Chang in UCSF, where they actually record from single neurons in the brain of human subject or patients, and they listen to sentences or to words, and collect activity of neurons, hundreds of neurons from responding to those speech signals. And we are able, in particular, we aim to understand whether we can actually find the emergence of a word manifold in deeper layers in this language brain area in the brain. And here, similar to vision, in the speech, a single word, the speech signal and the neuronal responses vary considerably depending on the gender and the speed of the speech, and et cetera, et cetera. So there is a manifold of representations of any one of the words, collecting all the utterances of the word, and we are trying to understand whether the concept of geometry of manifold applies here as well. And this is just an example of ongoing work where we analyze the same question, but in artificial neural networks that is speech to text. And again, on the y-axis is the separability of those word manifold. And as we can see here, again, there is the later stages of this language or speech-to-text network exhibit the nonlinear emergence of nice manifolds for words. So we hope that similar geometric ideas can actually apply to understanding language, not only on the level of word, but also the level of concept and sentences, etc. Okay, so my last topic is the spontaneity. And the hypothesis that spontaneity in the mind is the output of chaotic dynamics in neural networks, and it had been known for many years that recording from brain on different scales shows irregular rhythmic activity, which is not well structured in the level of spike trains, but also the level of EEG and even functional MRI. Okay, and where this variability or these temporal fluctuations come from has been the puzzle and the topic of study for many years. And in 1996, in a work with Carl van Bredswyk, we came up with a model, a very simple model, that shows that a very simple circuit motif, excitatory population and inhibitory population recurrently coupled and driven by external circuit can actually show chaotic dynamics, completely deterministic fluctuations. Those neural circuits generate strong excitatory and inhibitory feedback currents. The network dynamics in this case, as we showed, converge fast to a balanced state. And you can see it in the top right diagram, where the excitatory input, there is a very strong excitatory input fluctuation. There is a very strong inhibitory input fluctuating, but they almost cancel each other. So the net activity, the balanced state, is nicely fluctuating around the spiking threshold of the neuron. So strong inhibition, strong excitation, generated states where they nearly cancel each other. This is the balanced state, and these states naturally give rise to deterministic chaos or temporal fluctuations. Now, the original model was, as I described, a uniform model of one population excitatory neurons and a population of inhibitory neurons, and this is the way the chaotic fluctuations look like if you simulate such a network. But over the last years, we and others have studied extension of this for several populations by work with Jonathan Cadmon, but also more recent work about spatially structured connectivity. And this is work by Rosenbaum and Doron and their colleagues over several years. And interesting, they showed that spatially structured connectivity of inhibitory and excitatory neurons can generate under certain conditions not simply kind of salt and pepper type of chaotic activity, but actually traveling waves in irregular fashions, as you can see here. This, I think, we're at the early stage of understanding the dynamics, the conditions for such states, characterize them, but also understanding their function. So that's, I think, where we will be heading in next years in this question. So to summarize, excitatory inhibitory balance, first of all, yields realistic spike stochasticity as well as response diversity. There are, over the years, extensions and elaborations of the concept of excitation inhibition balance in terms of adding homeostasis and plasticity to make it robust. There are various flavors of excitation inhibition balance, more tight balance or loose balance, and importantly, imbalance between excitatory and inhibitory signals turned out to be a key circuit phenotypic factor or role in psychiatric and neurological diseases. So the idea that excitation inhibition are tightly balanced in the normal function of the brain is an important principle. Okay, so it has also been applied to certain artificial eye circuits, and finally connectomics, which we heard from Larry, beautiful implications of that in the electrical and the electric fish and the fly, has also in the mammalian brain has shown a remarkable specificity and tight EI control. And this is in this example. It is the recent work by Moritz Hemstadter and colleagues for a cortical column in the somatosensory cortex of the mouse. And what they found is that actually in each layer there are certain specific circuits which control precisely how excitation and inhibition converge onto a target neuron. So, by this, I would like to just add that I gave you, I think, a brief overview of circuit motifs of the mind in terms of the various types of attractor network, discrete attractors, continuous attractors, and how they feature in our modeling of working memory, long-term memory, spatial navigation, and predictions and planning, and some of the experimental validation of this concept. I talked about moving to higher order cognitive functions like discrete object, words, concept, action types, and so on, and how the geometry of manifold of neural representations in this particular case in terms of manifolds it turns out to be an important give us important insight and framework how to understand in modern language the sensory hierarchies, their function and their role in cognition. And finally, the spontaneous dynamics of chaotic neural networks, which are not representational per se, but they are an engines that allow potentially for creativity and spontaneous functions or aspects of the mind. And with this, I would like to end. I would like to thank Moser and Jayaraman and Burak and Lee for contributing to the presentation and for the support sources. Thank you.
Thank you so much, Haim, for that beautiful presentation. I guess you started with the provocative word mind, and so I'm going to press you a little bit on that, which is, I think, something we have learned, thanks to your work and many others in computational neuroscience, is that sophisticated computations can arise out of relatively simple mechanisms. And I'm wondering what you think about the emergence of mind throughout the animal kingdom. I think Larry showed us some examples where some of these mechanisms are present already, right, in quite simple animals. What do you think, is there any discrete gap between what we might think about as mind in the psychological sense and mechanisms that many animals have access to?
No, that's an important question, and I don't have a good answer or a good idea at which stage in the animal kingdom you say the appearance of the mind. So I believe that some of the mechanisms already exist in the fly. Maybe you can say that the fly is a mind. That's fine. But this is why, rather than giving a definition, what is a mind, I prefer to talk about a collection of functions or states and the underlying neural mechanisms. And then at what point it's rich enough to say, well, there is a mind or interesting mind. I don't know. I leave it open. We can discuss it later.
Okay, I have a quick question for Haim. I also agree, by the way, that the mind is a continuum, right? I don't think we should think of it as discrete. But in any case, So these attractors and manifolds are really exciting, and my recent work, in fact, is relative to your talk is in line, thinking about manifolds and traveling waves in cortical circuits. So I think that's really where the future is. But the question is, what's the dimensionality of these manifolds in the cortex? Can you put a number on it?
So I think the manifolds that are I would say well understood and simple enough, like ring manifolds and toroidal manifolds are I think well understood in terms of their dimension. The dimensionality of the manifolds that when we talk about objects, for instance, in vision, the dimensionality at the later stages are rather small, in the 10th. So there is an enormous reduction in dimension, which is expected. But when we talk about traveling waves, and I agree with you that your work on traveling waves is very much relevant to the recent attempts to model mechanistically both the emergence of those traveling waves, but also their function. The computational function is an important question, and I think it's largely something that we will see progress in the future, I believe. But these dimensions, I think, are relatively high dimension because they don't seem to have underlying regular structure. It's not a lattice. It's not white noise either. So they're intermediate, but I think typically they will be high dimension. But I think it's something that we should we need to understand better.
Haim, you gave this example of the language model reproducing or recalling this story. And of course, the impressive thing is it didn't spit it back verbatim, but gave an equivalent account. So I'm wondering, should we think of stories like that as a manifold of stories? Is that one way to think of how that kind of thing occurs?
Right. And that's what I showed the example of word, where the manifold has to do with the variability in the input signal, the physical input signal, like in vision. But we have another work which is purely language, where we are looking at the manifold of concept, and there the manifold is indeed has to do with the variability of the representation of the same word in slightly different context. So it's the same meaning of animal, but you have different animals and so on and so forth. And again, we have strong evidence that thinking about this as manifold is very fruitful. You can really take this and understand the progression or the emergence of the representation of that contextual representation in the network. So I really think this will be very powerful way to think about language representations.
Great, thank you so much, Haim. And let's turn to Terry's talk. And Terry will be talking about brains and AI.
Okay, let me get it set up here. Okay, well, first of all, thanks to the Lundbeck Foundation for making this prize possible. And I think the recognition here for the field really is enormously important, especially for the junior people coming up. And I have to say, there in one of the other Brain Prize webinars on learning and memory, I was really impressed with the 3 talks that were given. And I highly recommend everybody, if you're interested in learning and memory and synapses, plasticity, to go and listen to those talks. So I'm going to tell you a story that really begins back in the 1980s. And it was a time during which network models were being developed, learning algorithms for network models. Jeff and I worked on the Boltzmann machine. which was an offshoot of the Hopfield network, and both of whom, by the way, got the Nobel Prize, very well deserved, and developed tools for being able to create what we now call distributed representations. And I'll tell you a little bit more about what I mean by that. But using those tools, we created networks, and I'm going to give you a couple examples, which were giving us hints about how you could compute with distributed representations in large networks. And Pat Churchland and I wrote this book in 1992, which gave us a taste of that. And it was a time during which most of the recordings from neurons in the cortex and elsewhere in the brain were from single neurons, one at a time. And what we were doing is trying to develop a kind of a way of creating larger networks and thinking how single neurons could fit into them. Well, fast forward to 2012, and those networks, which started with just a few layers, developed into very large networks with multiple layers, and this led to the deep learning revolution that occurred in the 2010 to the 2020. This was a book that was published, both books published by the MIT Press, but it really, the tagline on the top is what you see here: artificial intelligence meets human intelligence. And now, of course, this is the way that artificial intelligence really has penetrated into the public. And I'll get to that at the very end. So, as I said, in the 80s, there were a really just an efflorescence of learning algorithms. And I just want to give you a feeling for one of them here. This is called blind source separation, or the cocktail party problem, which is you have multiple microphones in a room and several people are talking at the same time, and all of the sounds are superimposed. And the question is, how do you separate each individual source from the mixtures? And that's was a very difficult problem. It wasn't solved back in the 1990s. But Tony Bell, when he was a postdoc in the lab, came up with an algorithm, a neural network learning algorithm, which was iterative, like the ones today. But it was for a very simple network, which had inputs, which were the mixtures, and then the outputs, which were trying to separate the individual sources. And the algorithm was very simple. It was basically maximizing the entropy, which is the degree of complexity in the output, which would minimize the correlations and try to create independent outputs, statistically independent outputs. And that gave rise to the simple learning rule. And I'm going to demonstrate that for you here. This is for real data in a real room with two sources of sound, one coming from people talking and also from music. So that was one mixture, okay? And we had two mixtures. Now, after applying independent component analysis, this algorithm, ICA, here's what we get on the output. Well, okay.
Terry, were we supposed to hear anything? Because I didn't.
Oh, I'm sorry, you didn't hear anything.
No.
Oh, okay. I heard it, so I'm sorry that you didn't, because let's see, I don't know why it was suppressed. But in any case, what it demonstrated was that you could easily separate music from speaking. Now, that algorithm has been used in thousands of applications, and it's in the textbooks now for signal processing, right after PCA. And when it was applied to brain imaging, this is functional magnetic resonance imaging, it separated the resting state, where you ask a person just to sit there and to rest without any task. It separated out a number of different combinations of brain areas, which, remember, this is independent sources, but what they can be identified just from other sources of recordings as being auditory processing, language processing, and something which is very mysterious called the default mode, which had been discovered earlier by Rakel. This is just 3 out of about a dozen of these resting states, which are kind of like the joints of the different cortical systems. So I want now to take you back to the 1960s, and this is when Hubel and Wiesel made a great discovery in visual cortex, recording from single neurons, and most of what we know now really was derived from that era. What they discovered, recording from single neurons in the visual cortex of originally cats and then later monkeys, is that single neurons respond optimally to bars and edges in particular orientations. So here's recording the spikes. You can see as you rotate the bar, you get the maximum firing rate at a particular orientation here, centered on 0. And all many different orientations are represented in primary visual cortex. Now, these were thought to be edge detectors because obviously the edges were a very good stimuli, but it turns out that these same cells also responded to texture quite well. And it wasn't clear at all at that point, theoretically, what were these neurons representing? So Horace Barlow, who's a very distinguished visual scientist, suggested that this might be an efficient way of encoding natural scenes. Well, how do you test that? Well, here's what we did. We applied independent components to natural scenes like the one on the right. We cut out 12 by 12 little patches, as you can see here, from all over the image. And we used that as input to ICA. And the idea was, let's see if there are independent components that can go into reconstructing those patches. And indeed, here are those independent components. And you'll see that they have a remarkable resemblance to the properties of those orientation selective neurons in the visual cortex in different locations, different orientations, and also different spatial frequencies as seen in the cortex. The other thing, which is even more important from the perspective of efficiency, is that for any given patch, you only needed maybe a dozen of these in order to be able to reproduce the patch with some fidelity. In other words, it's a very sparse representation, and that is, again, what's seen in the cortex. Not all the neurons are activated, just a small fraction for any given input. Now, here's a second example, and this one now is an example of how, if you take a computation that you want to do, you can train a network to do it, and then you can compare the solution in the network to the actual recordings of neurons and see whether or not there's a correspondence. So this is a problem called shape from shading. And the question is, if you see a shaded image like the one on the bottom here, what do you see? Well, you see a little bump coming out. And you can see that it's illuminated from the side. And the question is, how would you compute that with neurons? Well, the first step is to have an input, which represents what is found in the retina. And there are 2 types of cells. There's on-center excitatory inputs and off-center inhibitory inputs. And what you're seeing here on the right is a diagram of the activity of the neurons in the model retina for this particular scene. You can see these are highly activated. The on-inhibitory are activated on the right. Now here's the network that we trained. We took those inputs and then we trained them on a lot of images like this one to be able to compute the orientation, first of all, of the surface. There's a long and a short axis, as you can see here. This one has the same size for both long and short. So orientation along the top axis, and then there's also the curvature, right? The curvature can be either convex or concave. It can go up or down. And so in this particular case, it's convex up, and it's oriented equally in both orientations. So we trained it up and we got these beautiful, it solved the problem. And now we can actually take a look, go into the network and look at the hidden units. And here's what we found. We found, first of all, that the hidden units, now, okay, I'm going now, these are representing the activity, as you can see now, the weights, the weights between one of the hidden units and the inputs. So there's excitatory weights to these inputs and inhibitory weights to those inputs. You can see this is an oriented type of an arrangement, and that's apparent in the output here, orientation selective. But what about this one here? This is also, by the way, these are called simple cells by Hubel and Wiesel. This is also a simple cell, and you can see horizontal orientation. But if you look at the output, it's not telling you anything about the orientation of the actual input. It's telling you something about the convexity or concavity. And so that's interesting. So what's the difference between these two? Well, okay, now there's something very odd about human visual perception. I think most people see at the bottom a crater. This is from the moon, but if you flip it upside down, it's an identical image, but it's upside down. You see a hill, and that's because you assume the light is coming from above, and it's amazing how this is a very discrete shift in your perception. Is very, very well established that you get these shifts. So what we did was we looked at the pattern of activity across all the inputs. The fraction of time in type 1 was active at a certain level. You can see is unimodal with the peak. There's usually relatively small activity levels. It's sparse. But the type 2 that I just showed you is bimodal. And that reflects the fact that either it's apparent to be either convex or concave. Now, here's the thing is that I mentioned they both look like simple cells, but how do you know whether it's a filter or it's a concavity detector? How do you know? Well, the only way you know is by looking at the output, which we call the projective fields. And this was actually in the title of our paper that appeared in 1988, which was the idea that the function of a cell cannot be determined simply by its inputs, which of course you can easily record, but by its outputs, which is very difficult to record. So there's actually no word for this. We created this word, projective field, and it's caught on. Now, the other example I want to give you is language, and this is now getting into the cognitive area, and Haim was already alluding to this. We now know that these deep learning networks they just love language. But we had a little taste of that back in the 1980s. This was a project it was actually a summer rotation project by Charlie Rosenberg. And the question was, can we train a network, just like we did with that previous example, to be able to correctly identify the sound of a letter, right? So C has two different ways of pronunciation. Could be the hard C in cat, or it could be the soft C in city. And it's determined by the context of the letters around it. So what we did is we took actually a text of sentences, and we ran them through a window and represented each letter by a unit. And then we trained it through a layer of hidden units to predict the sound. And some of these letters, like A, has a dozen different sounds. So it's a very difficult problem in English. And there's some rules. If you go to the phonologist, what they tell you is that there's hundreds and hundreds of rules in English, and there are exceptions to each one of the rules. And sometimes there are rules for the exceptions. I mean, it's 300 pages of rules and exceptions. So this is a difficult real rule problem. And unfortunately, you probably won't be able to hear it, but I'm going to show you what it sounds like as you start, and you tell me if you hear anything.
No, I do think we should be able to if it's just playing from your computer, because we can hear you. So
okay, then why not? I can hear it perfectly. But in any case, it's babbled. Okay, with the ba 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 ga da 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 ga. Okay. So it the first thing it picks up is a distinction between vowels and consonants. And then we played this. I'm not going to play it because obviously you can't hear it, but it actually is understandable. This is from a real transcription from a real human being. And so it was able to absorb both the regularities and the exceptions. And that was quite remarkable. Now, when you talk to people about these neural network models, by the way, this is a tiny model with 200 units and 20,000 weights. By today's standards, it was very tiny compared to these large language models with trillions of weights, or the brain, which has 100,000, hundreds of trillions of weights. So they say these are black boxes, but there's nothing stopping you from going in and analyzing it, and we did. So we clustered the activity, and surprisingly, maybe not too surprising, first of all the vowels here cluster together in terms of their activity patterns on the left, and all the vowels are here. Here's A, I, E, I, O, U, and sometimes Y, right? So these are the vowels, and here are all the consonants, and here's the C's, right? The hard and the soft C. So there's structure within the network, and you can actually pick it out. And now this is where we are today. I'm going to jump from here to these transformers, and just to show you that we can understand them perhaps in the light of the brain and how the brain solves language problems. So very, very, very briefly, these transformers, you get inputs coming in, and then they're fed into this decoder, and it's trained to produce the next word in the sentence, right? This is literally trillions of words that are being given. It's self-supervised. There's no labeled data here except the words coming in from the sentences, highly structured English. Now, what happens is that you ask a question and you get a word out, and then you take that word and you send it back to the input. There's a long input vector here that contains all the words that were given to it from the query, and also from the output, and then it just cycles one word after the next. There's a loop here, and this is a feed-forward network. This is just one of the blocks. You can have hundreds of blocks, and so how this is interesting. I'm talking now one word after the next. How is it that I talk? Okay, here's how the brain does it. Well, we have this motor cortex shown here for different parts of the body. And the motor cortex projects down to the basal ganglia just beneath it. It's topographically organized, as you can see from these colors. But then it loops back through the motor thalamus back to the cortex, loosely topographically. It's not one-to-one like the retina, but it loops about 100 milliseconds. And so this is producing one action at a time. In fact, the basal ganglia is important for learning what's called procedural learning how to string together a sequence of actions to produce something like learning how to play tennis or learning how to do mathematics. But it's you don't have conscious access to it, but you do from the cortex. You can talk about things declaratively. But what's interesting though is that you also can imagine learning through predicting what the next word is going to be. It might be that, as Larry was showing, predicting the next sensory input may be important, not just for fish, but also for humans. Now, the other thing that's interesting here is this secret sauce here in the transformer. It's called a multi-headed attention, self-attention. And what this does is it's a matrix that tells you how two words in a sentence are related to each other. For example, this may be related to a noun, right? A pronoun and a noun. And that's learned along with all the other weights in the network. If you ask where is there such a thing as self-attention in our brains, it might be down here in the basal ganglia because of the fact there's a lot of interactions occurring within the basal ganglia and a lot of learning taking place here too that's needed in order to produce smooth sequences of actions. Now, the last thing here I want to show you is that if you do this analysis of the hidden units, this is now in an early version of these large language models, but it goes back actually to 2013. Transformers were introduced in 2017, but already people had BERT and other they're called word embeddings. They produce word embeddings. You feed sentences, you predict the next word, and you get these word embeddings. Now, what was interesting when you analyze the hidden units, and this is now projecting many hidden units onto a two-dimensional principal component analysis, was that there are regions within this high-dimensional space of activity which cluster the names, as you can see, of countries. And there's another region here, in a different part of the space, which clusters the capitals of countries. That's interesting. So these are semantic patterns. So there's a semantic organization, but it's more than just semantic meanings of words. It also tells you something about the relationship between words. So just like Larry showed, if you look at vectors, if you think about vectors in the that represent things in the world, where the directions of wind and also your orientation in the world, you can also draw a vector in this semantic space between the country and its capital. And now if you take that vector and lift it to France, what does it point toward? It points to Paris. So these vectors are relationships, and it's telling you that there are geometrical relationships in this high-dimensional space that carry very rich information about how to represent words and relationships in sentences. And now that becomes really, really powerful with transformers. Now that takes me now to where we are today. We have created a bridge between understanding how brains work, recordings from the different parts of the cortex, and also artificial intelligence using very simplified models of neurons and learning algorithms. And that has created a new field. Next week, I'm going to be going to Washington. And the NIH has a meeting that was organized by some of the leaders in this area called Neuro AI, and it's the crosstalk between the people in AI who are trying to build these large language models on one hand, and also neuroscientists. And you heard from Haim about recordings from human language areas. We would like to understand, and there's a fantastic pipeline now between these two groups because they're using the same mathematical tools. They're collecting data and helping each other. And I think that's really a very, very important new development. Now, next month, in December, there's going to be an even bigger meeting that will be held up in Vancouver. The Neural Information Processing Systems, NeuroIPS, is having a meeting that will attract, it's the biggest AI meeting, it will attract over 16,000 researchers. And now this is really a field that has exploded over the last 10 years, and you read about this in the newspaper every day. This has really had a huge impact on the public. And just in the last 2 years, there's been an even bigger impact with these chatbots. ChatGPT, in particular, which was introduced 2 years ago by OpenAI, has really opened the eyes of the public on the capabilities of these networks. I have a book here that was also published by MIT Press. It was released just last week, ChatGPT and the Future of AI, the Deep Language Revolution. And what it tries to do is to help the public understand what's going on. They're confused. They see articles every day about they're going to lose their job. They hear about superintelligence. Regulation is really a popular topic, and it's very hard for the public to know what's going on. And this is written for the public, but it was written by an insider who actually can give them better insights, perhaps, into what really to expect in the future. Well, thank you very much, and I'm happy to answer a few questions.
Thanks, Terry. Maybe we could even take the questions from here to broaden out to a more general discussion. So I guess threading the loop between all of your talks, we've seen various examples of modeling that can account for neural behavior and behavior of creatures and humans from very small scales right up to human language. I guess I'm wondering what you think and others think about the role for more detailed biological mechanisms. We saw in Larry's talk that reference to the connectome and even to biophysical models, compound mental models, were really important to understanding the nature of the computation. That's something that's missing, of course, from from large networks. So where do you see us going in the future in really directly understanding the nature of biological brains? Do you think network modeling is enough, or that we really have to take another leap forward in trying to integrate that kind of biological information?
Okay, so I have a whole chapter in this in my book, and so first of all, you have to understand that deep learning is a model for only one part of the brain, the cortex, which is a very important part, and we all know and love it because it's our biggest knowledge store. But there are 100 other areas of the brain that are of equal importance for survival, and Larry really alluded to the fact that flies have very specific structures that are evolved to help them orient themselves. We have similar structures, and we can orient ourselves too. And he gave examples about figuring out where the door to the room is, but all of that is missing from these large language models. And we'll if you really want to be able to improve their performance, you're going to have to now add a basal ganglia. You really should add a bunch of other stuff too. Like I said, maybe it has the equivalent of basal ganglia, but I think going in the other direction, in my own research, what I've discovered is that these large language models have inspired me to think about what is the equivalent of the input vector, long input vector that is in the trans. It's very essential because without it's called temporal context, right? Like I gave you the example of the sound of a word, of a letter in a word, depends on the context around it. Now, if you're getting words in one at a time, you have to be able to have temporal context across words to understand the sentence. Now, the human doesn't have a perfect vector like a computer that accumulates, in the case of the transformer, accumulates tens of thousands of words, even beyond, practically a whole book. So how is that done? And I have a Trends in Neuroscience paper with Pat Churchland and Lyle Muller, former postdoc, suggesting that these traveling waves may in fact be the equivalent of a long input vector. And how that is done is very interesting. It's a new type of population code that we call the space-time code. So that just gives you an example of the back and forth between these two different approaches, the engineering approach to build something and the scientific approach to understand the brain.
I think, AI and neuros actually helps neuro focus on neuro in part. That the, at a conceptual level, the computations may be similar, but clearly the hardware is incredibly different. And I think in the past, we could have said, well, that's the only hardware, the biological hardware is the only hardware that can do these things. We now know that's simply not true. You can do it with another hardware. And I would say, for me, I still want to know how the biological hardware works. And now I have to be more precise about it because it's not unique. So in that way, clearly there's a synergy between the two types. But I think there's also kind of a mutual benefit in realizing the distinction between them, which has also become clearer.
I think first of all, from my perspective, I agree with I am on the Larry's side, that I'm primarily interested in understanding how the biological brain generates cognition and mind. But I think there is a real room for multiple scales of understanding, as already Terry showed in terms of spatial scale, but also in terms of temporal and complexity. More importantly, complexity. So, in examples that I've shown, it's kind of relatively simplified network motifs and how their power, but how they are actually tested or even implemented, if they're implemented in the real brain, I think it's an important question, and this requires to go more into the biological hardware, as Larry said. So I think these two directions, so-called top-down and bottom-up, should be working in parallel. But in my view, there is not really, in my view, it's not the approach, bottom-up approach, purely bottom-up approach is not viable. The complexity of the circuit of the hardware and the biophysics is such that it's hard to see, except for special cases, it's hard to see that we can gain insight about the function just from the bottom-up mapping. So we need hypotheses, either formulated in a qualitative sense like boxes and arrows, or preferably in terms of concrete models. But then these hypotheses and models need to be tested into whether the actual biology uses those mechanisms or those hypotheses to generate cognition or not. So, but a purely bottom-up approach, I think, is not the way to go.
So I think part of the reason why there's such complexity in brains has to do with evolution having another constraint, which is the amount of energy that is available to run the brain. Now, there's a big contrast here because, we run our brains on 20 watts. Some of us a little dimmer, perhaps, but these large language models take enormous amounts of power, megawatts. This is like, it's digital, so it generates a huge amount of heat. And evolution basically had to figure out very clever little, ways of not just having one solution fits all, but differentiating and having diversity so that you can have each specialized mechanisms, each specialized for a particular function. And because it's so diverse, it makes it more difficult, of course, to understand and decode. But I agree with Haim that, in fact, I think that both the bottom up and the top down are insufficient by themselves. And I actually, if you look at Mars diagram, what do you see in the middle, right? You see algorithms. So I think where we have the option, where we have, if we start and try to deduce what the algorithms are, and Larry showed that fortunately, evolution actually downloaded the algorithm into the actual connectivity, right? There it is. But understanding the algorithms that nature evolved will, with constraints coming from top down and bottom up, those are the ones that were really evolved to solve computational problems and survival. And so I think we're in a position now where we actually have enough constraints so that we can actually figure it out.
Let's come back to the issue of connectomics, actually, because, Larry showed some beautiful examples of that. And I think we've all at different times been supporters, to some degree or the other of that endeavor. How confident are you that connectomics is going to reveal in complex systems, in mammalian systems, the kind of algorithms that sort of appear and emerge so beautifully from the fly work?
Yeah, so I'll mention that. There's one thing that happens that if you've experienced it, as soon as you have a connectome, the way you discuss a circuit completely changes. Not that even in the fish stuff, we didn't even have the results of the connectome. We just had the pictures, and we were talking about it differently. And I think what people lose track of when they say, is the connectome going to give us the answer, is its ability to tell you you're wrong. Right? It doesn't isolate the right answer, clearly, except in exceptional cases. And those are going to be less and less in the complex systems. But, you might have thought, there's so many connections, you can write down any model and it'll be right. And I have personal experience that is just not true. In fact, the first 5 models you write down conflict with the connectome. So it's an incredibly powerful tool. And I think will continue to be even in more complex organisms for getting rid of the ideas that are wrong, at least, and allowing us all to focus on the ones that might be right.
But I agree with you. But I think if you go to more complex systems, mammalian brain, it's not clear what it means to be right or wrong. So let me give an example. Suppose I come up with the idea that in the case of self motion, the principle is cancellation of signals. Okay, and now the question is it really realized by the brain? And the answer is yes. Now, The connectome didn't, you can say, well, no, it's not in this stage, or not in that stage, and so on, but the principle still remained cancellation. So when you go to even more complex systems, it's not clear at what level of agreement you need in order to say that this is yes or this is no. So I think it is we need more, even more conceptual frameworks, even I would say, to think more about what we mean by that. So to me, connectome is, I think more broadly, connectome are extremely important for things that we may not care so much in our work. Development, evolution, genetics, health. I mean, survival. Cell types and the rules that makes the connections are all governed by different cell types and so on. So it's a wealth of biological information, which is extremely important. So whether we use it for our model or not, we should just make clear that I think connectome, whatever mapping it is crucial for the neuroscience and for health. But on the other hand, we are still, I think, in early stages of understanding what level of complexity that is revealed by the connectome already is important. In what sense? It's important for the computation. Particularly, we all know already that there are so many different inhibitory cell types in mammalian cortex. Okay? Now, how do we conceptualize, how do we go about dissecting their potential role in computation? I think this is one of the big challenges that we have now in the field. I don't have answers, but I think I would like to see more progress in this direction.
So Haim and Larry, this is really the crucial to understand, and nothing more fundamental, I think, than anatomy. And it's really interesting because it's descriptive, right, in some way. And in science, if you want to get a grant, you have to have a biological question, right, a hypothesis. And so connectomics doesn't have that. It just says, well, let's just build it, and and they will come, and we did. There was a famous debate that occurred, I think, at NYU between Tony Movshon and Sebastian Sion. Now, Sebastian, as you know, is one of the leaders in connectomics, and Tony is one of the leaders in visual cortex recordings and understanding something about, what different parts of the visual cortex represent. And I have to say that Tony won the debate in a sense that he really pointed out that if all you have is anatomy, it's really insufficient. But here's the way that I came away is that it may not be sufficient, but it's necessary, right? And so we have other sources of information. You want converging evidence. So, and a good example, and Larry knows more about this than I do. The flight, the direction of motion circuit in the fly eye, right? There's a lot more neurons involved there than people thought, in a very complex way. But it was, and this is something that Reichart had been working on, for decades and decades. And it wasn't until the connectomic and the tools for recording from those neurons became available, but it was the combination of the recordings and the connectivity that actually broke open the circuit. And so this is how we're going to make progress, right? You have to have multiple sources of data converging and ruling out all the possibilities that are. And by the way, there's more than one possibility. There's, if you look at different species, they may have come up with different solutions to the same problem.
I think, our backgrounds in physics really didn't prepare us for anatomy. It's kind of like when we came in, just give me the matrix and leave me alone, and I'll figure it out. But, because of evolution and development, it's clear that the anatomy is incredibly important. How the matrix got built is critical to understanding the matrix. So we've all grown into neuroscientists over the years.
So Larry, it's interesting because one of the big differences that I noticed early on, that distinguishes the biological approach from the physicist approach. If you do an experiment on an electron, at Harvard, you're going to get the same result as you would in Tokyo. And the reason is that all electrons are the same. In biology, there's nothing that is the same. Every individual is different. Every genome is different. It's just the opposite. Diversity is really a different set of principles. You have to think differently. And also, the concept of learning is not. You're right. Hamiltonian is fixed. If you figure out the Hamiltonian is, you can figure out you have a theory. But that's not true in biology because of plasticity and learning.
Larry, Terry, you're right. But there is something, the reason why I think all three of us are working in computational neuroscience is because there is diversity, as you mentioned, heterogeneity, enormous, but there is more or less uniformity or universality of function. We talked about mind. My mind is more or less working similar to your mind and to Larry's mind and to Adrienne's mind. There are differences, but first order, we have the human mind, and I think this is the most important motivation for looking or for hoping or for believing on principles which are more general than more universal than the enormous microscopic diversity. So yes, it's not an electron in physics, but nevertheless, cognition is so common, even across species, that there must be, in my mind, general principles that are realized in kind of different circuits, but the principles are similar.
Yeah, let me come back to that because we touched on it before, Haim, after your talk. One thing that I find very useful about our work in general is the idea that we have ways to pull apart, what are the mechanisms of various properties that people ascribe to mind, right? And I think you laid that out beautifully in your talk. Here's a list of things that one would ascribe to mind. How can they be done? You have a universal way of thinking about them that can be implemented across very different, even biological systems. So I guess what I'm wondering is what many people would like to know is when do we start to ascribe those properties to an artificial system? Because all of them are buildable, right? You can model each of those things. So what will it mean for an artificial system to have mind in the sense that we've been thinking about it?
So if I may answer, I think, as I said, mind has some properties or some modules and some motifs, which I think maybe not now, but there is no reason, to have. There are already multimodal systems and kind of AI robotics and so on. But I think that if I go maybe beyond the concept of mind, I think what is really missing now, in my view, is the agency, the aspect of agency. And the aspect of agency is not only, people say, well, you need a physical interaction with the world. That's fine. I mean, you can have AI robot that has physical sensors and physical motor actuators and a brain and mind inside. But I think one needs goals, long-term goals. And I think that is something that before we see kind of the independent agency, that I'm not saying it's impossible, but I don't think we see it at the moment in any of the AI systems. So I would say this is still something that is a huge gap between ascribing agency and whatever comes with it to AI, to current AI systems.
So Haim, that's a wonderful point. And again, it's a chapter in my book that AI needs goals. It needs a lot more. It needs a long-term memory, first of all.
AI has long-term memory. That's the easiest part.
No. Here, no. You look, if you talk to ChatGPT, and you know, you have a long conversation, you turn it off, and you know, it's gone. It doesn't remember it. Now it could store your whole conversation, but it doesn't. You can't just start up again. But no, that's not the point I'm trying to make. Here's there was a debate here at UCSD in cognitive science. This is just you know, within the last year, and the question was, does ChatGPT understand language? And you know, there's a debate format. You know, you go back and forth, and at the very end, they took a vote. And these are, you know, a lot of students and faculty. And half of them said no, they don't understand language, and the other half said yes or maybe. Right? And literally, these are experts. These are people that, you know, should know better. Right? If they can't decide. What it's revealing is that we don't understand understanding, right? If we can't conclude whether it is or not. And I pointed something out. I said, look, if you talk to a linguist, what they'll tell you is, Chomsky in particular, that syntax is really the key to language. It makes a difference between human language and animal communication, right? I'm not sure if I completely buy that, but one thing you can't deny is that ChatGPT can talk to you in perfect syntax, right? So if it doesn't understand syntax, how is that possible, right? So I think that we're at this point where I think it's revealing our ignorance, or it's asking us to think deeper about these words like consciousness, intelligence, and understanding.
I agree, but I think animals may have consciousness and agency without language. So I think language is important. I agree with you. Whether what language models have the understanding of language somewhat similar to humans is still an open question. But I think that they're not necessarily related, because you can have an organism, an animal, that has complexity of a mind and agency and consciousness.
We might want to leave it at that. We've probably used up our time. So thank you all so much for your contributions. And I hope that anyone watching will come to watch the rest of the series. So thank you very much.
Thank you.
Yeah, thanks. Great job.